Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Quick note before we begin, the Finding Genius Foundation, as part of the Finding Genius Podcast, has recently completed a book about understanding viruses. So the creation of this book was to interview 100 virologists, ask them a lot of deep, difficult questions, take the most difficult questions, and then re-interview the top 25 or so and ask them the hardest questions I could think of. And we compiled that all into a book. So you'll see question and 4 or 5 experts' answers. Question, 4 or 5 experts' answers. There's about 30 questions in the book. I think it's a great read for the layperson and for the researcher. It talks about a lot of speculation in the world of viruses, such as are they alive or not, and why is it important? Why do viruses go latent or hidden or ineffective or sit in a person or an animal or another creature for weeks, months, years, and then suddenly become virulent and affect that person? So there's a lot of really provocative questions in the book. It's now on Amazon. So if you go to Amazon and type in Finding Genius, you'll see the book on viruses. It's also on Kindle. The Audible version is in production and should be ready in approximately a month. But if you want to go and order it now, you can do so again by going to Amazon or Kindle, or go to findinggeniusfoundation.org and go to publications. There's an opportunity as well to get the transcripts of all the interviews and to hear the original interviews themselves. If we put them all together, the book would be about 1,000 pages, but we condensed them down to make it juicy and concise and tight and very interesting. So I hope you'll check out the book. We're now working on one about cancer, but this is going to be our goal is 3 times a year to come out with these masterclass books that I think will inspire new scientific research, and I hope you'll check it out. Thank you. Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. Before we begin, just a quick message. We have a new book called Understanding Viruses by Finding Genius. You can go to Amazon and type in Finding Genius to look at it. It's a compilation of interviews with 25 top virologists, asking them tough questions and for their speculative answers. So I think it's a good read. Check it out, again, on Amazon by typing in Finding Genius. Today, my guest is David Goode. He's a group leader, part of junior faculty at Peter MacCallum Cancer Center. And we're going to talk about his work. So, David, thank you for coming.
Well, thank you for inviting me.
Yeah, tell me about your research. What's it involved?
So I am a computational biologist. So that means my job is to analyze large, complex data sets related to cancer. And my group uses sort of evolutionary approaches to study cancer because cancer is a dynamic system. It's always evolving. It's always changing. Cancer cells have to be very adaptable. They face a lot of environmental challenges as the tumors form and grow. And then certainly during treatment, they're treated with drugs or radiotherapy. They have to adapt very quickly to survive under those conditions. And so we study these evolutionary processes. How will they help tumors grow? How they help them adapt, with a particular focus on the evolution of drug resistance. That is, the ways that cancer cells come up with to sort of beat the drugs that are used to treat them and continue to grow even after therapy.
Well, do you think that cancer is a separate life form with its own homeostatic drive and agency, or how would you characterize it?
Yeah, I wouldn't characterize it exactly as that, but I think that framework can be applied in many ways. Cancer cells are kind of like rogue cells in a way. They kind of break away from the constraints that are placed on most cells in terms of their growth and their spread. So kind of in a normal functioning multicellular organism like a human, there has to be a lot of coordination and cooperation between cells. You can't have cells just growing wherever and whenever they want to. But cancer cells, they kind of break the rules, if you will, and they start just growing and dividing kind of according to their own schedule. And of course, this is very damaging to the host. So you kind of have this competition, if you will, between the cancer cells who want to spread and divide and make as many copies of themselves as they can, and the host who's got all these mechanisms to try to stop cancer cells from doing that.
Well, do you think cancer cells act independently, or they act in concert at least in the context of, let's say, a tumor or metastasis?
Well, I think that what they do is they get some autonomy over their own growth and proliferation so that they kind of ignore the normal signals from other tissues, other cells in the body that would be telling them, now you should stop growing. They kind of ignore those and they just come up with their own means to keep going. But they certainly, they're not entirely independent. There's a lot of evidence that tumor cells interact with other cells around them in the microenvironment, and that they manipulate those cells to help them, help the tumor cells grow. And then that also different sets of cells within the tumor, different clones of cells within the tumor will actually compete, but also cooperate. So there's a lot of signaling that goes on between cancer cells and the cells in their environment, and between different kinds of cancer cells within the same tumor.
So when you talk about evolutionary effects, are you talking about just random mutation, natural selection, or does the evolution look different for cancer?
So a lot of the principles of Darwinian evolution certainly apply to cancer, that usually cancer is initiated by some sort of genetic mutation. Mutations are occurring in all of our cells all the time as they grow and divide. But if one of those mutations occurs in a gene that is important for controlling cellular growth, then that can then lead to the initiation of a tumor by sort of damaging one of the controls, that turning off a control that may tell that cell to stop growing, or the mutation may turn on a gene that tells the cell to start growing and dividing when it shouldn't. So there's certainly an element of Darwinian selection to that, in that you have these mutations that occur, and they can allow these cells to grow faster than the cells around them. And then so those cells are basically have a selective advantage in that they can outgrow their neighbors. So, and then, as the tumor continues to grow, it's kind of like a snowball effect. More and more mutations will accumulate, and there'll be, again, selection acting on those mutations that'll make some of these tumor cells, grow even faster than the other tumor cells. And so those ones will have an advantage and eventually dominate the population. So it's like a really dynamic system that's always changing.
So, after chemo treatment, it seems like tumors definitely become a lot more aggressive. Do you think that's just random mutation, or do you think it's deliberate adaptation in response to a stressor?
Yeah, so that's an interesting question. And I think there's sort of two possibilities that are not necessarily mutually exclusive in every case. And I think what I discussed about the sort of accumulation of genetic mutations and selection for cells carrying mutations that are advantageous, that help them grow faster, is kind of the traditional view of cancer evolution, and still a very valid view. And in the case of therapy, you can certainly have selection for cells that are more aggressive. They're the ones that are more likely maybe to survive the therapy because they can grow faster, they can adapt more quickly. But there's also accumulating evidence now that there are sort of non-genetic mechanisms of evolution in cancer. And these can sometimes be triggered by stresses placed upon the cancer cells by the environment, such as chemotherapy and other drugs. And that the drugs sort of have this unintended side effect of actually kind of making the tumors more plastic, more adaptable, and more aggressive, by kind of inducing them to switch on different genes and switch off other genes that actually make them grow faster and make them kind of better able to withstand the effects of the drug. So I think there's a bit of both. There's selection for pre-existing genetic changes, but then there are also changes that are induced by therapy that actually do make the tumor cells more aggressive.
So what are you trying to do? Are you trying to model how tumors grow, or their response to chemo, or what are you trying to do?
Yeah, we do some of that. We do, one of the things my lab is doing is trying to sort of computational simulations of tumor evolution under different conditions to try to figure out what makes some tumors more aggressive than others. And because, when you get a sample from a patient, you can do sort of DNA sequencing, RNA sequencing, you can look at what mutations it has, what genes are switched on in that tumor. But that's just kind of one snapshot in time. There's all these things that happened before the patient showed up in the clinic, and then there's all these things that are going to happen afterwards as that patient goes through surgery and treatment and so forth. And so to try to get a big picture of how that whole process is working, we can use mathematical models and actually simulate the growth of tumors in the computer and see how the different balance between the different mutations, between the different clonal populations in that tumor changes over time before and after therapy.
Okay. I mean, have you looked at a tumor 3D and map the heterogeneity? So maybe you can use a computer model to track it back to how it started or how many cells it started from. Have you tried that? Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit FindingGeniusPodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit FindingGeniusPodcast.com and click Support Us today. Now back to the show.
Yeah, we are working on ways to actually match our computational simulations to actual data. And we haven't been able to sort of use sort of 3D sort of heterogeneity data to do that yet. We're basically based that on the kind of genetic profiles of the tumors. So the numbers of mutations they have, how common those mutations are. We can use that then to estimate sort of how many different clonal populations there are and how common each of those populations are. And then we can see, okay, of all the sort of thousands of simulations that we've run, which one looks most similar to that particular
how are you
tumor.
doing that? Are you doing single cell sequencing, or how are you determining what mutations there are and how many there are?
So we actually just do that with what's called whole genome sequencing or exome sequencing. So it's not single cells. You basically take all the cells. You just take a sample of a tumor and take the DNA, and that DNA is going to be representative of a mix of cells in that tumor. So you're going to kind of get the genetic profiles of the most common cells in that tumor. The single cell technologies, I think, are really powerful, though, and that's something we are looking to move into, where you can actually profile individual cells. And so then with kind of more precision, at least in that sample of the tumor that you took, which clones were there and what their exact frequencies are, and you can detect sort of more rare clones. So one of the limitations of current techniques is that a clonal population, a mutation has to be present in about 5 to 10% of the cells. Otherwise, you won't know it's there. So that's
again, if you know how they're distributed throughout the tumor, I think that would give you a lot more information instead of just, there's 8% of this and 10% of that. If you look how they're distributed, if it's radially, or if there's a pattern to it, what would that tell you?
Well, the spatial patterns are very powerful in that they tell you kind of a little bit more about the history of the tumor, and that there's sort of different regions of the tumor might actually be genetically different, and they might be molecularly different. And that, I think, has big implications for drug resistance, because one part of the tumor might actually respond really well to the drug and sort of shrink and die off, but another part of it may be completely resistant, and that'll grow back. And one of the issues is when you have a patient's tumor, which could be quite big, you only take, when you take a biopsy, you only get a really small piece of that tumor, and so there's all this information about the surrounding tumor that you don't know about, and that can sort of mislead you in terms of thinking about what that tumor actually looks like genetically and what it's like molecularly, and you might actually miss some important clues about a better way to treat that patient.
What do you mean? Like, what would throw you off? What would you see that's not reality?
Well, let's say 20% of the tumor carries a mutation that would make it resistant to the drug that you're giving that patient, but that 20% is up in the sort of upper left corner, and you take a piece of the tumor from the bottom right corner and you sequence that, then you would have no idea that mutation was there. So that's one of the things that I think is really good.
So what does Histology do right now? What does Pathology and Histology look at? Do they do that, or how do they sample?
Typically,
is that even relevant to what you do?
Yeah, so typically they what's done is that you basically take a sort of a thin piece of tissue from the patient at biopsy, and that's basically like a sort of a needle biopsy usually, so a very thin sort of slice, and that's often what we get to use for our studies. Or sometimes we can get pieces of tissue from surgery, but you sort of again just sort of get a random slice, and then the rest of it will go for pathology, and they'll actually make slices of the rest of the tumor and they'll preserve it. But then for research we get sort of one of the pieces to work with. We have done some studies in prostate cancer where we actually managed to get preserved sections of a whole prostate. So when patients, some cases of prostate cancer, when it's particularly aggressive and spreads throughout the prostate, they'll just remove the whole prostate from the patient. It's called a prostatectomy. And we could actually sample 17 different regions within that prostatectomy sample. And we could see that there were clear genetic differences across the different regions of that prostate. And that's in line with a lot of other studies. So as you were alluding to, that kind of spatial heterogeneity is prevalent in cancer and is actually very important to understand. And so we're working towards ways to do that.
Okay. So what I mean, what would be your way of doing it? Again, is it single cell sequencing? But then you have the problem of, how many do you sequence? Hundreds, thousands, millions? What are your thoughts on how to do this in a better way?
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Yeah, so I think, again, single cell sequencing, you get better picture of the heterogeneity, but you lose the spatial information. You don't know where those cells came from in the tumor. So I think one of the sort of strategies that's important to think about when using patient samples is to try to sample multiple regions. Like I said, there's also new technologies for spatial profiling. There's something called spatial transcriptomics, where you can actually look at what genes are expressed in different regions of the tumor. So instead of the sort of typical approach where you take the whole piece of tissue and kind of mix it all up, you actually take a slice of it, and there's a technology that will actually allow you to sequence genes in sort of different parts of that tumor. And so that way you can kind of map that, okay, this section of the tumor has got these genes switched on, this section of the tumor's got these other genes switched on. And so we're looking to use those technologies to particularly to study prostate cancer, where we know there are these molecular differences between different regions of the prostate tumor.
So, I mean, what is this going to result in? Different chemotherapy protocols, or what do you hope to figure out by seeing this?
Yeah, that's a good question. I think partly it's sort of discovery driven, and that we don't know what we don't know yet. So I think that will be, that always will uncover new information, and from that you can get new ideas about treatment. I think, one of the things that I'm certainly interested in is why do these tumors become resistant? What changes have to happen before and during therapy and after therapy that make them change from a drug sensitive to a drug resistant state? And I think perhaps if we can look at things in a spatial perspective and we get a better picture of the heterogeneity and also the microenvironment around the tumor, we might give us some clues. We might see sort of cells in transition, or we might see differences between regions that tell us, oh, okay, that's why the tumor was able to come back, because in part of the tumor there were these changes that then became more common after the therapy. So see things like maybe mutations that are in one region. Well, then when the tumor comes back, those mutations are in every single cell, or the same thing with certain gene expression patterns. Maybe they're only one little part of the tumor has these genes turned on, but when you sequence it again after it's returned in the patient, you see those genes are turned in all the cells. So that can give us important clues about those evolutionary processes and how they're occurring and how that's leading to drug resistance. And then if we know that, then perhaps we can come up with better ways to treat patients that will prevent the tumors from becoming resistant in the first place.
But how are you going to see this? What method are you going to use, you think?
So you mean in terms of the sort of analytical methods or the analytical approaches?
Yeah, there's a trade-off between single cell and just wide swath of it. What do? You do multiple swaths? You do, multiple spots and then a couple single cells in each area? Like what would be a protocol that you would think would help you see in more detail what's actually going on?
I think for my work, it's really most important is to kind of have the sampling at different time points. So before and after therapy, so you can make those comparisons. And it's, as I mentioned, harder to do in patients necessarily. So
why is it hard? Why can't you ask for a resection of all, prostate tumors from a given hospital? Would they not give them to you, or is it what would happen with that? You
can get that if it takes some time and expense. It's more about the over time, though. I mean, if you have a patient, it's kind of hard to ask them to come in for a biopsy every month. You get poked every month. So what we do is we try to use model systems in the laboratory that are actually based on patient tumors. So one of the things that I work on very closely with collaborators is something called patient-derived xenografts, where we can actually take a piece of a tumor from surgery and then put it into a mouse and grow it inside a mouse. And so these are specially bred mice that have very weakened immune systems, so they won't reject the graft. And in that way, we can actually keep those tumors alive, if you will, in the lab in what we call an in vivo environment. So not in a dish, but actually inside a growing organism, the mouse. And we can see then how those tumors change over time as we kind of passage them from mouse to mouse. We can see how they respond to different kinds of drugs. And so that way we can get a much higher resolution picture of how those tumors are evolving than we can if we just rely on patient samples alone.
Have you done this yet or no?
Yeah, we're doing this right now. We have a big collection of different kinds of really aggressive prostate cancer growing in our mice in these xenografts. And we've started sampling them at different points in time. look at how their genetic profiles and their transcriptomic profiles change, we've started doing experiments where you have them in a mouse that is producing androgen. So prostate cancer relies on androgen hormones to grow, and that's mostly testosterone. And the main treatment for prostate cancer is to starve them of the testosterone. So it's something called androgen deprivation therapy that reduces the amount of testosterone in the system and thereby kind of slows the tumors down. But they always seem to find a way around it. They always seem to find a way to continue to grow, even in the absence of testosterone. And so we can simulate this process in the mice. We can put the tumor in a mouse and actually supplement it with human testosterone. So we know it has lots of testosterone. And then we can do what's called castration on another mouse. So we can take the tumor, put it in another mouse, and then deplete the testosterone in that mouse. And in that way, we can see how does the tumor change going from an environment that has lots of testosterone to one that doesn't. And so we're doing those types of experiments. And we're also now going to do experiments where we look at how do these tumors change when you give them chemotherapy. So we're going to give them a commonly used chemotherapy called carboplatin, and we can see how they change before and after. And for those experiments...
Are you just looking for genetic changes, or are you looking at epigenetics and microbiome and extracellular vesicles and all that stuff? It sounds like it's genetics and that's it, or no? Genetics
and transcriptomics for the most part is what we're looking at the moment. Yeah, we would love to do a lot more stuff. It's just there's sort of practical limitations and financial limitations on what we can do right now. But we're building towards getting a more complete picture. And this is actually where we're actively using single cell RNA sequencing, which you mentioned before, where we actually looking for differences between cells in the same tumor in terms of what genes they're expressing, what genes are turned on or off. And that way we can actually identify specific populations of cells that change in response to androgen deprivation or in response to chemotherapy.
Okay, so far from the transcriptomics and the genetic changes, have you gotten data back yet on how tumors change and after various chemo treatments and over time?
Yes, we've gotten some data back. We haven't gone into a lot of detail, so there's I can just sort of share with you sort of the high level things. I mean, a lot of the things we see in our tumors are, you get changes in the androgen receptor signaling in the absence of chemotherapy. So that androgen receptor is like a key gene for growth in prostate tumors, right? So it's activated in the presence of testosterone. And the tumors, though, will find ways to keep that signaling going even when the testosterone is gone. And another interesting phenomenon we see is that sometimes prostate tumors become independent of androgen receptor signaling, and they actually change the way they look. They change their molecular properties quite dramatically, and they do something called transdifferentiation, where they actually change into a different cell type. They change into from sort of an epithelial cell type to a neuroendocrine cell type. And that's a very interesting process. It's these tumors actually becoming something that's very, very different in response to therapy, and this allows them then to survive and adapt. So we can actually look at our models and kind of catch cells in the act of doing this. We can find populations that are in between the adenocarcinoma cell type they were before and the neuroendocrine cell type that they're becoming.
Yeah, why would they become the neuroendocrine cell type? What good does that do? Yeah. What abilities does it confer on the tumor?
Yeah, that's a good question. And I think there's a lot of research going on in that area. I think one of the key things is that by moving into that neuroendocrine cell type, though, they don't need testosterone anymore. So that's kind of like an escape mechanism. But why they do it that particular way is not entirely clear. But it's in line with sort of another sort of emerging viewpoint in cancer biology that tumor cells have this kind of what they call plasticity. So they have this ability to change forms. They're, whereas sort of like a skin cell or a lung cell or a bone cell will remain a skin cell, lung cell, bone cell, the tumor cells kind of go back into more of a stem cell-like state, and that allows them then to sort of shift their molecular profiles, shift their transcriptional profiles to become different things, and this makes them more adaptable. So a drug comes and, maybe some of the cells are susceptible, but if they can quickly switch on different genes and become something else, then that's a mechanism for them to survive treatment.
What about metastases in prostate cancer versus the primaries? What do they look like?
It varies again from case to case. I can't claim to be an expert at this point in metastatic prostate cancer, but you see a variety of different resistance mechanisms. But the main things again are they either change androgen receptor signaling by making lots of copies of the androgen receptor gene or mutating that gene, or they transdifferentiate into this neuroendocrine form. So there seem to be those two main pathways that tumor cells can go down.
But if you compare primaries versus metastases, maybe you'll see different things happening, different paths that'll shed light on what's going on.
Yep. And that's definitely something we're interested in doing. And we have in our xenograph collections, we have matched primary samples from the primary tumor of a patient and then samples from the metastasis from that patient, when they returned after relapsing, a year or two later. And so we can actually look at those in the lab and compare them and see what's changed, how are they different. And we've done this for one patient in detail so far. And it's interesting in that the genetic profile didn't seem to change that much between the primary and metastasis, but the metastatic tumor had all these different genes that it would switch on, but only in the castrate host, so only when there was no testosterone. So when there was testosterone, the tumors had some differences, but they had more differences when the testosterone was gone, and there were more genes, kind of more growth pathways turned on in the metastasis. So again, this is sort of pointing to the cells in the metastasis sort of being more adaptable and being able to switch on more types of genes to help them survive.
Is the goal of therapy, or is it just understanding? I mean, what do you hope to understand about these tumors? And then what therapy would that lead to? Or is this more just, you're happy with the understanding of what's going on with them?
Well, we want to move beyond that eventually, but I think there's a lot of complexities to understand first before we can, definitively say a particular therapy would work. But the idea is to see what's different about the metastases in the primary, and then to identify potential drugs that we could test. And a good thing about our system is that because we have those tumors growing in the mice, we can actually test drugs immediately in the same tumor. So if we identify something that looks like a molecular vulnerability in the metastasis, then we can actually go ahead and test that with a specific drug if the drug exists to target that pathway.
Are you going to be allowed to test any drugs? Or because from what I understand, the standard of care has to be done first through sections and chemo, and then things can only happen kind of after that, but not in the absence of it.
Yeah, so that's true for patients, but not in the lab. We have these tumors growing in the mice, so we can test all different kinds of drugs because it's an experimental setup, right? So if you're not actually testing in patients, you have more leeway to use different compounds, even sort of experimental drugs that aren't used in the clinic yet.
Yeah, but if you do that and you get a result, are you going to be able to surface it, or again, are you going to have to only do a clinical trial in which the standard of care is used first?
I guess I couldn't give you a detailed answer on that. I mean, the sort of part of the standard research process is you try things out in the lab first and on cells and on mice. And then when something works, you then look into carrying it forward into the clinic to test in humans. And, first you've got to show that it's safe to use in humans. And then if it is, then you test it in cohorts of patients where some get the standard of care and some get the standard or get the new treatment. So it's all part of the process. We're kind of in the, discovery phase, you could say, trying to find better drugs for prostate cancer because the current therapies don't work that well.
What about the prostate-specific antigen test? How does that correlate with mutations and, the morphology and the mutation heterogeneity in the tumor?
I couldn't actually really speak to that very knowledgeably about PSA. I know that it's used to monitor basically disease progression in prostate cancer, but that it has its limitations. My research is really more around looking at these evolutionary processes and this plasticity and, as we call it, this ability of tumor cells to sort of switch states. And so one of the reasons that we think that tumor cells can become more plastic is actually that they kind of regress from multicellularity into a more unicellular state, which is another sort of big topic of research in my group.
So is the epithelial to mesenchymal transition just another stage in this de-differentiation? Like if you compare that process to just, inside of a tumor itself, the de-differentiation, is one more marked than the other? Are they similar, or is one more than the other?
You're saying, are there differences in the level of differentiation in cells within the same tumor?
Yeah, like, if a tumor is preparing to send out cells to create metastases. Yeah. From what I understand, I may be wrong. They undergo a transition where they de-differentiate a bit. Then they're able to slough off and move around and become metastases. But does that happen to all cells in a tumor, or just certain ones that appear to be destined to go out and travel? And are there spots in the tumor that the de-differentiation, like if you were to characterize the level and intensity of de-differentiation in a tumor, what would you see? Has anyone looked at that?
So I'm not sure if that exact study has been done. I think, though, you would see a lot of heterogeneity in the level of dedifferentiation. And you certainly will, you can see in certain types of tumors, maybe some cells will be undergoing the epithelial mesenchymal transition and others won't. And that you'll have various stages of dedifferentiation within the same tumor. And I would certainly propose that the cells that are less differentiated, they've kind of gone further back towards a more stem-like or more adaptable state, would probably be the ones that would be more likely to metastasize, because again, they would have that ability to be more adaptable to new environments, which you think, right, if this tumor cell is going to metastasize, it's go in the bloodstream, which is can be quite a harsh environment for it. And then it has to go to a different tissue type and colonize that. So it really has to be very, very robust, very resilient, first of all. And then it has to be very adaptable. It has to be be able to quickly adapt and change to its new environment. So
well, I
how
wonder,
do they do that?
If a cancer metastasizes, the microenvironment of the metastases would be a lot more alien and harsh than the primary.
Yeah.
So I wonder if there's less of a redifferentiation when the cells arrive. I don't even know how you characterize it, but I guess just like characterizing genetic heterogeneity, how do you even characterize the degree of differentiation or dedifferentiation?
Yeah, that's a good question. One of the ways my lab does it is to actually look at what we call signatures of loss of multicellularity. And we kind of have a model where the tumor cells kind of transition from being more of a differentiated multicellular type of cell to something that resembles more a unicellular organism like a bacteria or yeast. And we can study this process by looking at how old the genes are that the tumors are expressing. So we can compare the sequence of human genes to hundreds of other species, and we can identify genes that are shared with very primitive single-celled organisms like bacteria, like yeast, genes that are very, very ancient. And then we can also identify genes that are only shared with other mammals, and those are much more recently evolved. And what we see is that tumor cells, they shut off the more recently evolved genes, and they upregulate, or they increase their expression of very, very ancient genes that are conserved with bacteria and single-celled eukaryotes, and also genes that are what we call early metazoans. So they're genes that evolved early on in the process of multicellularity. So our hypothesis is that tumor cells are their transcriptomes are kind of being changed. They kind of rewire them to move back into something that is more of like a more primitive, more adaptable cell type, something that's a bit more like a unicellular organism. And so this then allows them to become more plastic, to become more adaptable, and actually become more robust and handle the stresses in their environment better. Yeah,
it's kind of strange that they do that. Do you know if metastases, again, if you looked at it again in terms of the origin of these genes that are being turned on, I don't know, what do the most ancient pathways look like? Or when does the regression or the atavism happen most strongly? Does it happen in metastases or in tumors that have been treated with chemo? Where does it happen most?
Yeah. So we're looking into that. We do see that when you compare tumors at early stages of progression, so low-grade tumors are kind of benign neoplasms that have not yet sort of fully become tumors, and you compare them to high-grade tumors, very aggressive, faster-growing tumors, that when you compare these sort of early tumors to later-stage tumors, you see the later-stage tumors have turned on many more of these ancient genes, many more of these highly conserved genes than the early-stage tumors. So that suggests as the tumors are progressing, they're shifting more and more into more of a unicellular-like state. They're losing features of multicellularity, becoming kind of more unicellular-like, and that this may be helping them progress and adapt. And so we're actually now following up by trying to look at what does the profile look for metastatic samples, or what does the profile look like after treatment. And so that's work that we've just initiated. But I would guess that this process of sort of shifting towards more of a unicellular-like state would continue and be enhanced in these metastatic tumors.
Has anyone tried to, I don't know if you could do this, but, take two tumors and seed one of them with cells from a metastases, or, take two tumors, treat one with chemo, and then see their progression, but seed the untreated one with cells from the treated one and see if that alters the trajectory of that one. So, the whole reason I'm asking is maybe there's some cell-to-cell signaling going on that governs the rest of the tumor. Part of it's instructing the rest to dedifferentiate or to do other things.
Yeah, that's an interesting idea. There have been some really fascinating studies conducted where they have done something similar to what I think you're suggesting, where they take different tumor clones, so different tumor cells from the same cell line. So they're actually from, they have the same origins, but they have been engineered to have different types of mutations. And when they can put those cells into mice, they do the xenografting like we do with the patient samples. They can take those tumor cells and put them into mice and watch how they grow. And they found that there's often cases where when you put two different types of cells, two types of tumor cells that have different mutations, they grow better together than they do on their own. So there certainly is some sort of cooperation, or maybe it's the competition or something that actually helps them become more aggressive and more metastatic when there are a greater diversity of clones there. And so, why is that? I don't know if that sometimes maybe they're making factors that help each other out. Maybe this is sort of more intense selection when you have more clones competing for the same resources. So it's kind of that iron sharpens iron idea. But there's those dynamics they definitely matter. That level of heterogeneity within the tumor and which clones are there and the different proportions. So there's some interesting studies done, particularly in breast cancer and in lung cancer that are trying to look at those questions.
Okay. Well, very good. Dave, what's the best way for people to find out more about your work? Where can they go?
I guess they can go to a website at the Peter MacCallum Cancer Center, and we have sort of an overview of the work we're doing there and links to some of our papers. And that's probably a good place to go for information. Yeah. And if you want to get in touch, all my details are there.
Okay. Very good. Dave, thanks for coming. I really, really appreciate it. It's been a good call.
Well, thanks for your time too, Richard. And glad for the opportunity to tell you about my research.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.